Okay. Good morning, everyone. I am Mona, and this is our team sitting there. So we choose this particular topic: optical magnetometry of single biocompatible micromagnets for magnetogenetics application. So there can be multiple applications of diamond and cell based magnetometry. We choose this one particular idea, how we can genetically modify or manipulate biological micromagnets at microscales using NV centered magnetometry tool. So going to the motivation, actually, so there are many cellular processes which are on the scale of micrometers, and if we use magnetic nanoparticles of the similar size scale, and we can create, we can manipulate the magnetic nanoparticle distribution around the cell, inside the cell, either through force, temperature, friction, by the external stimuli of magnetism, then this would be a non-invasive manipulation at the cell level. So this is a good application. Secondly, the challenge is that these kind of modification require magnetic field gradients of a large scale, are of the order of 10 to the power almost 3 Tesla per minute. So this is a large gradient. And these nanoparticle motion is also sensitive at millitesla scale uh, to the amount of magnetic field strength. So we need to make a setup which is where we can simultaneously control it, and also we can characterize the distribution of magnetic field strength and gradient. So actually magnetic tip is a good setup because these are generally in the range of say 40s, 50s nanometer at distance from this magnetic nanoparticle cloud. This magnetic nanoparticle can be like any general magnetic nanoparticle, but enclosed within a biocompatible material, which could be silicon based gels, or there are other materials also like PDMS. So they don't react with the tissue cell the same way as it would be otherwise. They allow the passage of gaseous molecules, so this way they will not hinder the usual cell functioning. If we enclose a magnetic tip very close to such a system, it can create this much gradients, and it can also drive the magnetic nanoparticle distribution around it. But the problem is the characterization. So to characterize the qualitative value of field gradients and magnetic field is difficult in such a setup, because the characterization of tip itself is not easy. So what is the distribution of magnetic field gradient, or the size, or I should say the magnetic field magnetization of the tip itself is not easy to characterize. But this would be very interesting if we can remotely control the magnetization this way, and we can manipulate the cells. So we are proposing, so I will tell first how we can create a system with these high magnetic field gradients, and what is our tool other than the conventional magnetic tip spectroscopy. So we can create these high magnetic field gradient system by making magnetic microparticles. So this is just a simple scheme. For this particular paper, they use photolithography technique, because the size of micromagnets we want to draw is of the order of hundreds of micrometer. Even tens of micrometer we can easily grow with this photolithography technique. But if we want to go to at a smaller scale for particular applications, then e-beam lithography is the best method, because at that scale we can fine tunely precisely control down to 50s of nanometer in practice. So the method is simple. We'll take some biocompatible substrate, or we can make it biocompatible after from the top side, so it can be either bottom top. We are using a cover glass leaf, then we deposit copper on it. This is a technical requirement, because depositing iron or any alloy on top of it will facilitate the deposition if we have a magnetic metallic system underneath, because the method used is generally electroplating or some kind of sputtering. So if we have a metallic layer underneath, it is easier to do that. Then we mask it. So mask is, as the name suggests, so either it can be a physical mask, you have some certain sort of patterns like square, rectangle, whatever you want, or it can be computer control, that you give your system a certain software, some pattern in the design, and then it will laser track where the UV exposure need to or not. So why I am saying that, because basically this is some sort of resist. Resist is a polymer which is sensitive to the UV radiation. So depending the exposure, time of exposure to UV radiation, how much soluble it will be in a certain kind of developer solution will change. So we are using positive resist in this case. Positive resist means if we expose certain area of that resist, it will easily dissolve in the developer. So whatever part I want to deposit, I can just simply let that open. So in this case, let us suppose this is my mask, I expose the UV radiation. Because these regions are exposed to UV, when I will put it in the developer, this resist will dissolve easily, and after then when I will sputter the magnetic, so in this case it is an alloy of nickel and iron, permalloy which is called, so it will deposit here. After that, this alloy can be washed away using a different solvent, so that dissolving solvent depend on positive or negative resist, what kind of we are using. Now we can also remove the copper underlith. It is kind of etching technique which is also used in the PCB industry, so it is standardized actually. And on top of it we deposit PDMS. So PDMS is a polymer layer, so it does not react with the tissue cells. It allows the passage of gaseous molecules, and it also does not attract or attract or repel the molecules by itself, only under the influence of some stimuli. So it is a good standard to use. So in our case, we have fabricated this kind of array of square micromagnets with the size of around 100 micrometers, and at the edges of these each micromagnet, we have a gradient as desired for our application. And the tool we used is the NV center optical magnetometry. So I can skip this slide, or I can just brief, because Professor Rock has already explained it to us in detail. So we have ground state and excited state, which is spin split because it is spin half system. Now under the application of magnetic field, we can even further Zeeman split the M plus minus 1 state, and the excitation of this will be beneficial, because the fluorescence either from 0 state or plus minus 1 state is actually different. So we can sense the change in fluorescence if we do a transition from lower to higher state, and that we can do because this energy of the range of this transition is actually of the order of gigahertz range. So we can do that through external microwave excitation. So we can flow a high current through a coil, simple coil around the setup, and it will generate the microwave, and that can undergo this excitation. Once the system excites, then we will see that there is a change in the fluorescence observed, because the number of population at the 0 and 1 state we have switched. Now the interesting thing is that this, because this is a spin split system, so if we apply a magnetic field, then this position of this peak is also sensitive, because now it is not just 0 to 1 transition, depending on plus 1 or minus 1 separation, it will change. So this way we can sense actually the magnetic field surrounding the setup. For the NV center system we are used, as we discussed in previous class, we have different orientation of nitrogen. So orientation of the vector along the nitrogen atom and the valency axis. So I am just giving it orientation. This is not the lab frame, this is the material frame. So let us say I gave a magnetic field along any direction, then there will be different components around 1, 2, 3 and 4 say. Because in the experimental frame there are different magnetic orientation, so projection of magnetic field around each will be different, which means the strength of magnetic field experienced by each configuration will be different, and therefore therefore the fluorescence spectrum will show a characteristic different peak around it. Now this I said only for the case of like one NV magnetometry. Actually we are using it with the purpose so that we can expose a large fraction of area in a single shot experiment. So that will give us benefit that we will have a higher resolution, because the resolution, the signal to noise ratio here goes by the square root of n. But at the same time, because we are sensing it through a camera and then we are analyzing it pixel by pixel, so the size of pixel will limit our resolution. That also depend on the wavelength we are using, but that is fixed for our case, and also the aperture of the cell camera. So in this particular configuration and this work I am going to tell, the resolution is limited to 500 to 800 nanometer scale. So, oh. So, how we do it actually? So, instead of using a tip diamond cell at the tip, we actually are using a film of diamond where we have deposited a large implantation of nitrogen vacancies. So, it will have certain thickness of few micrometer, and near the surface we have a distribution of nitrogen vacancies. So, we have a wide distribution of different orientation. Let us say I use for 100, then we will have around equal probability of getting it around 111100. So that we have a distribution of NV vacancy axis. So this is the setup. So what we are, so the benefit is that because the diamond film is CVD grown and it is polished optically, so when if we apply a laser pulse, it can go on multiple internal reflections such that it reach the NV layer, and from the NV layer it will get the fluorescence signal which we are detecting by passing it through objective lens to narrow it down. And this diamond cell is used as a probe, and this micromagnet system is our design system. If we want to study certain cell system, then we can put it in between and we can see with and without the cell how the magnetic field distribution is modified. And this micromagnet has a benefit permalloy that it can be easily magnetized, and also the magnetization can be switched easily using any external magnetic field, which is using in our case. So for this particular work, we need at least 3 different orientation NV spectrum resonance. So let's say we did it on 3 different direction. Now after that, to get it in the real frame, we use the maximum probable outcome methodology. So it's an algorithm like we have 3 different direction and we'll see in what is the maximum probable distribution which will give the compatible spectrum outcome for this. So this way, we can actually extract the X, Y and Z component of magnetic field in the lab frame. For this particular model, actually now I mentioned the micromagnets are of the size of hundreds of micrometers, so it may predict that the system should have quadrupoles or a complex anisotropic magnetic field distribution. But it turned out that for this system, the macro spin model can easily explain this. So that actually depends on the thickness of the micromagnet film we are depositing, and also the actually it also depends on the intersize between the micromagnets. So if we reduce it, there can be coupling on the adjacent micromagnet that can make the distribution complex in the each micromagnet. Like if we grow a smaller micromagnet size of let's say 50 to 100 nanometer, we will see a prominent quadrupole like signature. Okay. So now this one slide I just showed to say that whatever I were proposing as a motivation is actually can be used as an application. So it is shown that these are 4 micromagnets and we have deposited certain cells around it and a distribution of magnetic nanoparticles. The magnetic nanoparticles are enclosed in the silicon gel, and by application of magnetic field along different directions. So initially we have no magnetic field and the magnetic nanoparticles are surrounded, and this is just the cell structure. When we apply magnetic field along a certain direction, at the edge the gradient is large. It can drive the magnetic nanoparticles in motion, and these magnetic nanoparticles can enter the cytoplasm cell. And when we turn off the magnetic field, the motion can be otherwise, because still we have the gradient of the same magnitude in the opposite direction. So this way we can non-invasively drive the magnetic nanoparticles in the biological system for the application we were proposing. So I think I already exceeded the time, so I will just stop.
Okay, thank you very much.
Can we ask one? So if I come back to the image of the magnetic field, this one, yes. What would be the, or
magnetic field resolution actually can be very good because
What set the limit of that? So here somehow I see something which is as a function of there is lack of phase dimension. So my question was simply what is you see missing here? So everything is exempted around 100 micrometer centimeter.
so what sets the limit of magnetic field?
The resolution.
Resolution of magnetic field. Actually, in the CVD layer, we have large distribution of NV and center ions. So, if there is large cluster, then the time, the decay time of the resonance in the peak, that will modify. So, how that will change, I have to go, but I think that can contribute to it.
So for instance, how does what's the influence of the distance between the NV layer and the, 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 the magnetic sample?
Yes, so actually the magnetic sample, magnetic field, stray magnetic field decays exponentially with the distance. So if we increase the distance between them, then the stray magnetic field strength will drop to microteslas order or even millitesla.
Yes, but
Then
also now suppose I have 2 points, okay, that produce, so like, it's difficult, but two very, very tiny dipoles, magnetic dipoles, each of them will produce a given magnetic field distribution.
yes, yes, yes.
So suppose now instead of having these large microdot, I have very, very tiny one. To what distance can I put them as close as possible and
That
still being able to see that there is indeed two and not one?
okay. So that depends on the
So what
dipolar
I will call
strength.
a kind of spatial resolution. You see, my spatial resolution, I would define it as I have two dipoles, very, very tiny. I approach them, and until what distance I can do my measurement and still say reliably this is two dipoles.
That is actually limited by the interdipole interaction. Actually,
No,
you're right.
no, it's not limited.
No.
Actually, it could be the distance of the NV layer.
Yes, so I actually assume that NV layer is near the surface in
Yeah,
this
but
case.
if it has to be as close as possible. If now I remove it, you see that somehow I will broaden the
Ye-
magnetic field. This spot will be broadened,
Okay, okay.
and somehow at one point I will average the two response of
Yes,
the dipole.
yes. So it is the depth of the NV center ion inside the
It's the distance between, not the depth. The depth is very close. It's 10 nanometer from
10
the
nanometers.
surface, but the distance between the sample and the diamond and the sample.
Okay.
You see what?
Yes, yes.
Yes.
So, but
And of course, it depends a little bit on the signal to noise ratio, because it's somehow it's a combination of this parameter and the signal to noise ratio. Because if I have a very good signal to noise ratio, I can always do a kind of fit that will tell me it's not two dipole. So it's like the Rayleigh criteria. The Rayleigh criteria, you say it's lambda over two, or
yes, yes,
okay,
light
but it's
effect.
lambda over two provided that that was by height somehow. With a detector, you can go better than that, depending on your signal to noise. So it's the same idea.
So we can control the thickness of PDMS layer if we want to tune it.
So one of the limits of this method, a practical limit, is you want to have you will win a lot, a huge, in terms of sensitivity, resolution, everything will be much better if you are able really to push the diamond plate really against your sample. And
Okay.
this is not so easy because you always have some particles or whatever, and that will somehow. So there, in our case, we are limited to 30 micrometer just by pushing it, and that was the point.
Okay.
If you take care, you can go to 2 to 3 micrometer
Yes,
on the
actually,
deposition.
right. So in this case, it is I think order of tens
Exactly.
of micrometer, which is large.
Exactly. So the
Okay.
sensitivity of this wide field measurement is what is very important. It's the distance between the diamond, the NV layer that probes the magnetic field, and the magnetic sample itself. But
Okay.
you can achieve amazing really very sensitive measurement provided you're close enough, and very high resolution also.
The
Maybe
spatial resolution
for
will.
example, one can do NV in the cell, then maybe you have much better
Yes, yes.
satisfactory. Yeah.
But in that case, the idea was that they had no mean. When we were approached by the police, by these people, they asked us if you come back to attack.
Yes, yes,
You're perfectly explained.
yes, yes.
Congratulations, it was very nice. So no, this not this one. The very, very first one
Cell
is the
on the.
motivation when you see the cell.
Okay, okay.
Yes, this even no, no, it's the first one. Yeah. So for that, I mean, they had these microdots, and the idea was to use them as a kind of drive for the nanoparticle, as you explained. So they had to quantify the magnetic field that was produced by lab and see whether or not it was in agreement with the motion that they observed.
Okay.
And actually, it worked. But they had no way to measure the gradient of the magnetic field. So they went, they did a lot of trial measurements, and when we did that, we immediately we could provide them with the answer, and they were very happy about that.
So if we do the scanning method, there we sense the force between the tip and sample.
Yeah, but yes, sure.
So in that case, I think we can get a qualitative idea of the motion.
Congratulations, very good.
Okay, thank you very
Thank
much.
you.
Who is the next person? We can have maybe one more and